Then let's dive in with sensations, which is still going to be on the nervous system. Thinking back to what we talked about on Tuesday, we really were discussing motor aspects of our nervous system. Okay. Remember the title of that last lecture was somatic and autonomic, which are divisions of our nervous system. Somatic, which is responsible for controlling our skeletal muscle. Autonomic, which is associated with controlling our cardiac muscle, our smooth muscle, and our glands. Okay. When I use the term motor, I can also use the term efferent, because we learned earlier talking about nervous tissue, those terms are pretty much synonymous, and refers to signals going away from our central nervous system, either the spinal cord or the brain, out to those effectors, muscle, glands, that kind of stuff. Okay. This lecture, as you can see, is labeled sensations and the afferent nervous system. We kind of can use the terms afferent and sensory interchangeably, which is the exact opposite, really, of what we talked about last time, which is where signals are going away from the spinal cord and brain. This is where signals are going to go towards the brain and spinal cord. So the first thing I want to just do is just define sensation. Which is the conscious or subconscious awareness of changes in our external or internal environment. What do I mean when I say conscious awareness?
Like we can feel it without happening.
Yeah, exactly. Like what you're hearing me say right now, what you're seeing up on the board, what you're feeling like in your hands, when you're itchy, when you're tickled, when you're hot, when you're cold. Okay, that's all perception. Perception you usually can see, and this is not the first time we've seen this definition. Conscious awareness and interpretation of sensations primarily is a function of our cerebral cortex. Okay, remember in our lecture on the central nervous system, at least if you don't remember now, you better remember by next week, we talked about things like the primary visual area, primary auditory area, primary gustatory area. Those are specific areas of our cerebral cortex dedicated to processing those sensory signals. Okay. Do we have conscious awareness of every sensation happening to our body? Can we feel like when our pH in our blood is 7.35 instead of 7.4? Do we feel when our calcium levels are changing in our blood? No. Okay. We have subconscious awareness of those things, which means we really don't have, any idea that they're really being, things taking place inside of us. Okay. The nature of the sensation, the type of reaction generated vary according to the ultimate destination of nerve impulses conveying sensory information to the central nervous system. What that sentence there really means is kind of just these different sensations are going to end up traveling to our central nervous system. It's where they end up going kind of really depends on or determines whether they are going to allow us to have perception or not. If it goes to the cerebral cortex, places like those primary motor or primary auditory areas, primary visual, gustatory, whatever, we have conscious awareness of it, but we don't have conscious awareness of it because they don't go to our cerebral cortex in some cases. Now, regardless, each type of sensation, what we call a modality, like touch, pain, vision, or hearing, we group them into two categories, the generals and the specials. Okay, we'll spend about half of our lecture really just dedicated to the 5 special senses: smell, taste, hearing, balance, and vision. And then we will spend not quite as much time talking about the general senses. There's actually 2 types of general senses: somatics, which really they always say come from our skin, muscles, tendons, joints. Okay. So examples of somatics are the tactiles: touch, pressure, vibration, itch, and tickle. The thermals: warmth and cold, pain, And proprioception is that conscious awareness of our body's position of muscles, joints, tendons, which is, involving stuff we've actually talked about back when we discussed reflexes like our stretch reflex when you tap on that patellar tendon. So we'll be talking more about spindles and Golgi tendon organs and that kind of stuff. We won't really get into visceral senses, which provide us information about conditions within the internal organs, whether that's like the stretch of a blood vessel, tell us like blood pressure, pH of our blood, O2 levels in our blood, pH in our stomach, stretch in our stomach. Those are quite a bit more complicated. But we'll talk about, things like that when we get into the various organs like our stomach, our, respiratory system and stuff like that. Not so much going to get into them in this lecture. Now, regardless if it's a special sense, if it's a visceral sense, if it's a somatic sense, they all begin the same way, which is stimulating a specific sensor receptor. In this case, we show a unipolar neuron. Remember, unipolar neurons have their cell body off to the side. The receptors are the dendrites of that unipolar neuron, sensitive to cold. Okay. Now, not always are dendrites the sensor receptors. Sometimes we'll see specialized cells, like in our tongue, for example. We have special cells called taste cells that then synapse with the dendrites of what we call first order neuron. And the general purpose of these sensor receptors, regardless if it's a specialized cell or the dendrites, is just to convert the energy of a stimulus, in this case cold, into the energy of a nerve signal. What I mean is creating nerve impulses or action potentials so that we can propagate those to our central nervous system, and then we can interpret that stimuli acting on and within the body. So we decide what to do with the cold information that's being sent to our central nervous system. In the central nervous system, the process of converting stimulus energy to nerve energy is called sensory transduction. So transduction, or to transduce, means to convert, as it states here on this next page. All right, let's talk about the 4 events that take place for a sensation, or when a sensation occurs with the body. First thing is we have to stimulate that sensor receptor within what we call its receptive field. Okay. So reading through stimulating the sensor receptor, or stimulation of the sensor receptor, an appropriate stimulus must occur within the sensor receptive field, the body region where stimulation activates the receptor and produces a response. I always kind of use the analogy of like my wingspan. Okay, if I pretend that, if I stand up and I got my arms spread wide, okay, neurons are, like me, and I can pretend that I'm stuck in my position that I'm in right now. I can't move. Okay, if Logan was to throw me a ball, I would only be able to catch it if he threw the ball within, my wingspan. If he threw it over there by Lauren, I couldn't catch it, could I? Because like a neuron, I'm not able to move. And, well, you can't stimulate a neuron if you're not stimulating it within its wingspan, within its reach. Okay. Well, the wingspan refers to how spread out the dendrites are. Okay. Now, another example of this would be like we have only the ability to see certain amounts of, the world that we look out at, right? Can we see behind our head? No, because that's not in our visual receptive field. Our visual receptive field is from the very left side of where we see all the way across to the right side. We can see those things, because we've got the ability to carry that light into our retina, we can't carry light from behind us unless we turn around and put our eyes on it. Okay. Now we'll talk about receptive fields a little bit more here in a bit. We have to then convert or transduce the energy of that stimulus into a graded potential. Remember, a graded potential is also known as a local potential. It's a small depolarization. And if a specific strength of that stimulus is met, we will then create action potentials that can be propagated. What am I saying when I'm talking about strength and if we meet it? That letter T that begins the word spells out threshold. If we hit threshold, meaning if we depolarize enough that we hit that, what was the number, remember? Threshold is what it is. What's the number? -55. And that's when the floodgates open, in which our voltage-gated sodium channels open up. And then the voltage-gated potassium channels open up. We repolarize all that stuff that you guys hopefully know by now. And if you don't, you better know it by Tuesday. Okay. So really what I just was saying is step 3. When the graded potential in the sensory neuron reaches its threshold, it triggers one or more nerve impulses, which then propagate to the CNS. These neurons that propagate the impulse are called first order neurons because they are the first in a line of neurons. The ones they synapse with are called second order neurons, and then you've got third order neurons and so on and so forth. Okay. Eventually, we make our way into the central nervous system where we can integrate it. A specific or particular region of our central nervous system receives and integrates. Integrate, think process. We process that sensory nerve impulse and decide what to do with it, if there's really anything that we need to do. In this case, it looks like we're just taking like the back end of a pen or a pencil, pushing it on your skin. Is that going to really require any response out of us? No, not unless this was like super pointy and it hurt, or if it was super hot, which means that we're going to activate that flexor withdrawal reflex, like when we stepped on the tack that we talked about in our last lecture. So a lot of times we just send that signal up to our cerebral cortex for perception and say, oh, I feel that pen or pencil pushing it on me. No big deal, not an important piece of information. Because if you think about it, we're constantly being bombarded with sensations. What we see, what we hear, what we smell. Do we have a response for every single one of those things? Not really. For most things, we just, we take it in and that's it. And a lot of times we just filter it out. We talked about how you can hear noises outside, but your brain doesn't even really pay attention to them. You just kind of filter them out. All right, what we're going to do for the next couple slides is talk about the different types of sensory receptors that we have. There's a few ways to classify receptors. Okay, the first are based on what they look like under a microscope. That's what these 3 classifications are: free nerve endings, encapsulated nerve endings, and separate cells. Okay, and here's what they look like. The first 2 are essentially the same thing. Free nerve endings and encapsulated nerve endings are only different because with free nerve endings, the dendrites are uncovered, whereas with encapsulated, there's a capsule around them. Okay. So free nerve endings are described as bare dendrites, having no special association with specific accessory cells or tissues. They're very abundant in our epithelial and connective tissues, places like our skin, for example. They include our warm and cold receptors, opposite one another. Warm respond to rising temperatures, cold respond to falling temperatures. Nociceptors are pain-sensitive receptors. I always tell students, most people, if you ask them, do you want to stimulate your pain receptors? What would their answer be? No. So that's an easy way to remember it. I say most because some people like pain, but I won't get into any of that. And then hair root plexuses, which we talked about in anatomy. Remember, for those of you who are in anatomy, those wrap around your hair follicles and enable your hair to essentially be a sensory receptor, and why you can feel when you touch your hair even though your hair is dead. Okay. Encapsulated nerve endings are just nerve fibers wrapped with glial cells. Remember, those are accessory supporting cells of the nervous system, or just connective tissue. Function of that is to increase the sensitivity and specificity of the receptor. So it helps us with, it is easier to create an action potential. So we hit the threshold easier, and we're able to pinpoint where that sensation is actually taking place. Which I guess I put that on there twice, the exact same sentence. Whatever. Separate cells, like what we show here with a gustatory or taste receptor, are cells that are first excited, causing them to release neurotransmitters that then activate the first order neuron. So basically we've got a cell that then synapses with our neuron here, whereas here we're directly stimulating the first order neuron. Same thing here. So these guys, I always like to call them special cells. We don't have a lot of them. Really, they're actually called separate cells. Now, this is important because these sensory receptors produce two different kinds of graded potentials. Remember, a graded potential is that small change away from resting membrane potential that, if strong enough and it hits threshold, we then create nerve impulses, action potentials. These two, free nerve endings and encapsulated nerve endings, both create the same thing, what's called a generator potential. A generator potential, when it reaches threshold, triggers one or more nerve impulses in the axon of that first order sensory neuron. The resulting nerve impulse propagates along the axon into the central nervous system. So it's called a generator potential because they generate action potentials. Receptor potentials are what we do with our separate, or like I say, special cells. Okay. We still end up the same way. Action potentials are nerve impulses propagating into the central nervous system, but there's a couple extra steps involved. First, we have to activate the separate cell, which triggers the release of neurotransmitters. And remember all those steps of synapse transmission we talked about. The movement of the vesicles to the cleft receives the neurotransmitters cross the cleft, they bind receptors, all that wonderful stuff we talked about in our first lecture of the last unit. That's all taking place here. Notice we've got synaptic vesicles. Okay, there's the cleft, the space between the cell and the dendrite. The dendrite would be where the receptors are for those neurotransmitters. Okay, so receptor potentials trigger the release of neurotransmitters via exocytosis of synaptic vesicles. The neurotransmitters cross the cleft, produce a postsynaptic potential in the first order neuron. This PSP may then trigger one or more nerve impulses, which will propagate along the axon into the central nervous system. So really the kind of moral of the story is the same. Action potentials being propagated into the central nervous system. It's just we have a couple extra steps to go through, some more hoops to jump through to get to the end goal. Now, like I said, we don't have a lot of these separate cells. Examples would be in our ears for hearing and equilibrium, because both of those sensations take place in our ears. Hair cells are what they are called. What you saw in that picture was a gustatory receptor cell or a taste receptor cell. In our tongue, those types of cells are found in our taste buds. And then in our eyes, photoreceptors, like what we call the rods and cones. Anybody ever heard of those before? Well, we will talk about them towards the end of the lecture. Okay, so that's the first way to classify receptors. The next way to classify them is based on what they detect, the stimulus that is that they detect, which can be in the form of mechanical energy like a sound wave or pressure change, Could be electromagnetic energy, like light or heat. Could be chemical energy, like in a molecule of glucose that we taste, whether that's on our tongue or we actually taste it, and I said that really loosely, in our blood, so that we know we can regulate our blood sugar. So the classifications of these, the way to classify them this way are into mechanoreceptors, chemoreceptors, thermos, noses, photoreceptors, and osmoreceptors. Mechanoreceptors, called that because they respond or are sensitive to mechanical stimulation. Mechanical stimuli include things like deformation. What do I mean when I say deformation? The beginning of that word spells out deform. If something's deformed, it's not in its normal shape, right? Okay, think about like if you push it on your skin with your thumb. Okay, we've got cells that respond because they have changed their shape when you push in. When you stretch your muscles and your tendons and that kind of stuff, you're deforming them and the cells that make them up. So that's how they can become stimulated. Stretching, like in the stomach or in your muscles, bending of cells. So they provide us sensations of touch, pressure, vibration, proprioception, as well as hearing and equilibrium, as well as monitoring stretching of blood vessels and internal organs. Chemoreceptors, key here is sensitive to chemical changes, either the amount or the changing of a concentration of them, either internally or externally. In our nose, that's what we're responding to when we smell things, chemicals. We taste things, those are chemicals. We also, and like I said just a little bit ago, we can kind of taste our body fluids. I say that really, really loosely. So it's not actually taste, but we have the ability to monitor blood pH and blood glucose, blood CO2, blood O2, those kinds of things. Okay. Thermoreceptors are activated by changes in temperature, both warm and cold. Nociceptors, I already told you guys, those are going to respond to painful stimulation. Things that can cause pain would be physical or chemical damage to tissues. Photoreceptors respond to light stimuli that strikes the retina in our eye. And then osmoreceptors are activated by changes in osmotic pressure, which, if you want to think of it this way, it's just simply like water levels. So if that helps you.
So like when you get like when I get flushed my port, I could taste like fluid that they put in there. Why is that? It was gross.
You don't know.
When you get your IV flushed, you can always taste it. It tastes like rubbing alcohol or something.
Yeah, I don't know. So I've heard of that. That's a good question. I don't know.
It's probably just because it's going, into your body, into your bloodstream. Because even like when you get a CAT scan, when they put your contrast in, it feels like you pee yourself, like all the way down. You can just feel it because it's like rushing through your veins.
Which there's also what they call synesthesia, I believe is how it's pronounced. I'm thinking it. I'm 95% sure that's what it is. Where some people experience like really weird things with their sensations. It's really cool, but like it's just all about how we interpret it in our brain. How people can like almost mix and match their sensations. Like they can like trigger tastes when they see a color and stuff like that. It's really cool. If you ever want to go down a rabbit hole, check out synesthesia. You'll be amazed. A third way to group sensory receptors is based on their location, specifically of the receptors in the origin of the stimuli that activate them. There's what we call exteroceptors. Think extero for exterior, which is where they're at, either near or at the exterior of our body. What's the most obvious external part of our body? Our skin, right? Okay. Skin is also known as your cutaneous membrane, which is why you'll notice there behind the exteroceptors, it's listed as cutaneous receptors. Their job is to be sensitive to stimuli originating outside the body and provide information about our external environment, things like pressure, touch, pain, and temperature. Infra would be the opposite of those. Interoceptors are located in our interior, also known as visceroceptors because they're located within our viscera, which are our internal organs, blood vessels, the heart and the stomach and the intestines, so on and so forth. Muscles.
Are your internal, like organs and whatnot, are they more sensitive than like your external? Because I know it sure hurts more than like my husband goes stumps and stuff. It's like a whole different kind of pain than like.
Yeah, we'll get into like differences in sensitivity. I don't know specifically like internal versus external. I know like different parts of our skin are more sensitive than others, but we'll get into it. We'll explain.
Your armpits versus your.
I was about to say,
I
I
don't
think
know.
We'll get
arms,
into it. I promise.
face versus your.
We also have receptors actually within the nervous system themselves, or itself, because we have to monitor conditions within, like our brain and, the cerebrospinal fluid in there as well. So interoceptors are going to provide us with information about our internal environment, things like pressure, stretch, chemical changes, and even hunger and thirst. Proprioceptors are located in muscles, tendons, joints in our ear, provide information about body position, muscle length and tension, and the position and movement of your joints. Proprioceptors are like the muscle spindles that we talked about the other day that were activated when you tap on the patellar tendon. They were also the ones that were involved in the tendon reflex when we want to reduce tension at the muscles facing. There's two types. We'll talk about tonic and phasic, what those terms mean, but they provide positional information on the body or body parts while at rest or during movement. So table 16.1 here just kind of reiterates those three bases of classification: microscopic in the three types, receptor location, stimuli, the three types, and then the six types of, or six different types of stimuli detected groupings. Nothing brand new or anything there. Looks like do you need to write that word out there at the top that's cut off? Okay, I'll just do this. Can you read that?
Yeah.
Receptive field. So I've already kind of started to talk about this, but an important aspect of a sensory neuron is its receptive field. What constitutes its receptive field varies depending on the type of sensory neuron involved. We can see examples of different receptive fields at the bottom. I used, like my wingspan as an example, because I think, the stretch of my arms, it's kind of like the stretch of a neuron's dendrites. You can only catch the ball if you throw it within, my wingspan. You can only stimulate a neuron if you stimulate it where its dendrites are. If you try to stimulate it out here, it can't catch the ball. Okay. I also use the example of what we see, what we, can perceive only is in front of us, not behind us, because everything outside our visual space is not going to, interact with the dendrites in our eyes. Okay. Other examples would be like the amount of chemicals that we need to experience a smell, amount of things that we need to, as far as chemicals go, to initiate a taste, and sound waves as well. Considering all the different types of sensations that our body experiences, the receptive field of every sensory neuron can be defined as the stimulated physical area, like what was shown here, specific groups of chemicals like smell and taste, or particular sets of sound frequencies. Regardless, they have to cause a response in that neuron or neurons. In our somatic sensory and visual systems, receptive fields are physical areas, and receptive fields for neighboring sensory neurons may be in separate locations, like we show here, where they are not overlapping, or they can be like here, where they are overlapping. And this is super important, okay? If sensory neurons have separate receptive fields, then a given neuron will respond only if there's a stimulus present in the receptive field associated with that neuron. And so like what we're showing right here, I'll just, name them blue and green. Okay. Here's their receptive fields shown with the circular white areas of the skin. Notice we see no response in our green neuron, but we do in our blue neuron. Okay. It's like throwing the ball over to Lauren, but not to me. I can't catch the ball if it's not within my receptive field, can I? Okay. So we see action potentials being generated in the neuron that has a receptive field around the stimulus, or the stimulus is within that receptive field. Okay. So if sensory neurons have separate receptive fields, then a given neuron will respond only if there is a stimulus present in the receptive field associated with that neuron. If sensory neurons have overlapping receptive fields, now look at our purple and pink. Even though if we were to separate pinks by itself, the stimulus is not happening within its receptive field. Okay, because its receptive field will be right there, and the stimulus will be right over there. But look, we still are creating action potentials. Not as many, though, as what we see over here. So if sensory neurons have overlapping receptive fields, then all participating neurons will respond to a stimulus, which extends into the region of the overlap, but the response of each neuron is proportional to the relative position of the stimulus. Since the stimulus is more closely associated with the purple neuron, we create a lot more action potentials, meaning we have a higher frequency of them than here. Now, why is that important? Because it helps us pinpoint. Like if you were to push in on your skin with a pencil, you will be able to locate it more easily. This is what we call stimulus localization. Presence of overlapping receptive fields improves stimulus localization. I'll get into that more here for a bit. By contrast, when a sensory neuron has a separate receptive field, our brain has no way of knowing exactly where in that receptive field the stimulus is located. There are no signals from other sensory neurons that can be used for comparison. Meaning that this one, we really could be stimulating it anywhere within that little white circle, and our brain would not have any idea where exactly it's taking place. Might not be a big deal, but it could be a big deal. So it could be coming from here, or over here, over here, over here, over here, over here. We have no idea specifically where it's happening. Now, places where we need to have really great pinpoint action, great what we call localization, we see a lot of overlap between the receptive fields of these neurons, especially in our somatic sensations like touch, pressure, itch and tickle, and vision is a really important one. This enhances our ability to localize somatic sensory and visual stimuli. As you've already learned, sensor information is ultimately converted into action potentials before it's conveyed into the central nervous system for integration. If all action potentials are the same, how does the nervous system tell the difference between one stimulus and another? Meaning, how can we tell if it's an itch versus a tickle, whether it's hot versus cold? Because the signal's coming in as an action potential the same way, regardless if it's hot, if it's cold, if it's pain, if it's pressure, if it's smell, if it's taste. So how the heck does our brain know, oh yeah, that itches, or ow, that is hot, or this is tickling me? This is accomplished by what we call sensory coding. Using organizational and functional features of the nervous system to represent specific details about a stimulus, and we encode 4 attributes regarding a stimulus, or attributes of a stimulus, the modality, so what the sensation is, where it's coming from, location, how intense of a stimulus it is, whether it's like a firm touch versus a light touch, whether it's extreme pain versus light pain, and the duration. If it's continually acting on the body, or if it's a short-term sensation. So we'll start with labeled line coding, which relates to modality. So we said already, and that first sentence is exactly the same sentence that was in the very first page of our lecture, or I guess second page if you don't count the first title slide. Each type or unique type of sensation, such as touch, pain, vision, or hearing, is called a sensory modality. So this is talking about how we can tell, smell from hot, from cold, from pain, from pressure. Okay. We encode, or modality is encoded by which sensory receptor and neural pathway are activated when a stimulus is applied. The different types of sensory information that we have are conveyed to different parts of our cerebral cortex for perception. This relates back to that lecture when we talked about the cerebral cortex and the primary visual area, primary auditory area, primary somatosensory area. All those different sensations are not going to the same place in our brain. When we see something, that's being processed in our occipital lobe. When we taste something, that's going to be processed in that part of our cerebrum. Versus when we are touching something, it's going to that part of our cerebral cortex. And we also use different sets of neurons. I mean, it's not like the same neurons carrying the information for touch as they would for taste. Okay.
Well, that's just good. It seems like the symptoms of COVID and it's really just taste. I don't think that.
I don't know. I have no idea, to be honest. But yes, I've heard that. Smell and taste, I guess, COVID has affected. Neural pathways that convey information about a modality from peripheral receptors to specific regions of our cerebral cortex are called labeled lines. Here we show 5 different examples of labeled lines. Okay, a set of neurons that transmit information from only one modality, taste and touch and vision and hearing, or it could be balance. In this case, it would be hearing, because from the coat cliff, it was from here would be balance. And then smell. Okay, those are just 5 examples. Okay, but they carry the information for only that specific sensation. And this is why we're able to, see with our eyes, hear with our ears, feel with the pressure on your arm, because we stimulate those receptors in each of these parts of the body. And we activate a specific set of labeled lines that carry that sensation to a specific region of our cerebral cortex, where we then perceive the modality associated with that labeled line. The association of a modality with the activation of a particular labeled line is called labeled line coding. So we've talked about coding, talked about labeled lines, so when you combine those, we call it labeled line coding. Continuing to talk about location, let's talk about acuity, kind of one of my favorite things to talk about. Oh, and you know what? I forgot. I always have students do this. So it's going to be kind of trickier this semester with masks on. But take an extra pen or pencil. And I tell you, we won't do it behind our ear like I normally would do. Just put it in your non-dominant hand. And just hold it. And don't let go. If you don't have one. We'll get into why I'm having to do this in a little bit.
Are we gonna hold it for like a minute?
You'll hold it for a while.
I'm not using turn my light.
Acuity is described as the sharpness of perception. In other words, this relates to the ability to precisely locate and distinguish one stimulus from another. So to figure out where we're being stimulated. Okay. In our somatic, sensory, and visual systems, 2 major factors affect acuity: the size of the receptive field and what we call lateral inhibition. Okay. Let's talk about size of receptive field. which is demonstrated with the figures here at the top. Okay. A way to illustrate this process is by doing what's called a two-point discrimination test, where you've got a caliper, looks like a little compass, where it's got 2 points at the end, and you can change the distance across, so basically how far away the points are from one another. Okay. Well, look at the size of these 3 receptive fields with respect in comparison to these 5. And I know the picture is kind of small, but hopefully you guys can all see that these are bigger and these are smaller. What's something else that you notice right off the bat? These are overlapping and these are not. These are separated. Okay. The size of the receptive field varies inversely with the number of sensory receptors that it contains. So when there's only a few, they're going to have to be really big. To cover more ground, to cover more surface area. Okay. The smaller the receptive field, the more densely packed they are with one another, and that increases acuity. The larger the receptive field, the less densely packed it is with sensory receptors, and that lowers its acuity. Okay. Once again, acuity is the ability to precisely locate and distinguish one stimulus from another. Looking at this illustration, with our caliper, both points are pushing in on this singular neuron in its receptive field. Since it's only pushing in on this receptive field, our brain will actually interpret that as 1 point touching the skin, despite the fact that there's 2 points touching the skin. Whereas over here, the calipers, you can see there's a tip here and a tip here. Would be able to tell and can feel the 2 independent points. I wish we had time, because this is a pretty fun little experiment. I have these kind of calipers where you can actually spread across the points from one another, and you can test different parts of the body to see where the most acuity or the best ability to localize is. And where do you think we have the most sensitivity at?
Like where we would feel the most, or like where we would hurt the most or something?
Well, either way.
Like probably like your hands and your feet.
Why do you say that?
I don't know.
Okay, so if you were asked, okay, so I have young children. Okay, one of the first things that we do is, with kids is, read to them books that have, the stuff that they can touch, right? You can feel like the soft duck and the whatever. What's a baby or what's a kid going to feel? Those, their hands and their fingers, right? When you touch something, when you want to feel a texture of something, what are you using? Your hands. You're not going to walk up there and rub the back of your arm on it. If you did, you're kind of weird. You're not going to rub your calf and say, oh man, that feels so nice and soft. You're going to walk up there and you're going to touch it with the palm of your hand and your fingers. Okay. We also have really great acuity in our lips and our tongue. Exactly. They're feeling it. They're touching it. Okay. Look at this graph right here. Okay. Basically, the smaller the blue line, the better and more acuity it has. The index finger is the most sensitive, because that's what we touch things with. We feel with our fingers and our lips. And you said the toes, your feet. So we're making contact with the outside world with those things. You're not rubbing your arm and your calf on something and saying, oh, that feels so nice and soft. Maybe you will to be a weirdo, but not naturally.
Just whenever you're
Look at where they were measuring this 2-point discrimination test at. Tip of the finger versus your back. We don't need a super sensitive back. We need super sensitive fingers. At the bottom, we show what's called lateral inhibition. where we're pushing in on the skin with a pencil. Okay. Notice we've got 3 sets of neurons, a green, a purple, and a blue. Okay. We're most stimulating the purple one. Everybody agree with me? Which you can see we've created the most frequent amount of action potentials, the higher number of action potentials. We are stimulating the green and the blue because as you see they overlap, but look at the number of action potentials we're creating there. We just talked about that back on this page, right? We're stimulating more frequently here because we're more stimulating that purple neuron than the pink neuron. Okay. What's also taking place later on down the line is lateral inhibition, where what we see actually, the neuron that's stimulated by the side neurons, the green and the blue, they're actually inhibited because of branches from the middle one. So what ends up traveling to the central nervous system is a not very high amount of action potentials from the outside ones, but more so from the inside middle one. And once again, that helps us tell exactly where that tip of our pencil is pushing in on our skin, so we can, if needed, move that pencil away from us to protect us, get us from getting hurt, right? This stuff is super complicated, but hopefully you guys are seeing how neat it is, though, at the same time. Good to see you guys are all still hold your pen or pencil. All right, let's talk about stimulus intensity and encoding it. Question for you: how can your sensory system detect stimuli at differing intensity if all nerve impulses are the same size? Okay, meaning that if it's a really soft touch versus a much more firm touch, the action potentials that are going to be sent into our central nervous system are the same size. Remember, positive 30, 35 is where an action potential tops off, right? Doesn't matter if it's a really strong stimulus or it's a, right at threshold stimulus. Okay. Why does the light touch feel different than one from firmer pressure? Main answer to these questions is the frequency of action potentials. Frequency is referring to how often they are generated, so how often we fire the action potential. Okay. So what we show in our little kind of graph over here are 2 different stimulus intensities. The blue boxes down here at the bottom are showing you the strength of the stimulus. So this is weak compared to this that's really strong, okay, which results in different sized receptor potentials. With a weak stimulus, we don't depolarize a lot, but we depolarize enough that we hit a threshold enough that we fire off some action potentials, but especially look at these last few where they're spread out. Compared to over here where they're really, really close together throughout, they start to kind of taper off and get a little bit farther out. But here they were really spread apart compared to here. The more often you send an action potential is a direct result of how much of a stimulus strength there was in the first place. So our brain receives more action potentials from a firmer touch than from a weaker touch. And that's how we can feel different stimulus intensities. Why we can tell if it's a firm touch versus a light touch. On top of that, and I really like this illustration, really demonstrates it really easily. Look what happens when you initiate a really strong pushing on the skin. It can activate, in this case, 3 different neurons, whereas here, just pushing in very lightly is only going to stimulate this one. So a second factor is the number of neurons recruited or activated by the stimulus. Firmer pressure, for example, stimulates a larger number of neurons than light touch. Stronger smell is going to stimulate more neurons in our nose and a weak smell. A strong, itch is going to stimulate more neurons than a weak itch. Make sense? We experience different intensity of sensations all day, every day. And our brain processes that. All right, adaptation. This is probably my favorite thing to talk about in this lecture, which relates to the duration in which the sensation is acting on the body. So stimulus duration is encoded by the duration of the action potentials in the sensory neuron. In general, the longer the stimulus lasts, the longer the sensory neuron produces action potentials. Meaning that, if something's acting on the body, our body's going to create action potentials for that amount of time, right? You would think, not always. Okay. If a stimulus persists, like that pen or pencil that was supposed to be held in your hands, okay, you felt that pen or pencil in your hand right away, right? Okay. After a couple of minutes went by and I started teaching and you were writing things down and you were listening to me, were you focused on the pen that was in your hand? No. When you got dressed in this morning, everybody got dressed this morning, I see clothes on you guys. You feel the touch of that shirt on your skin, whether it was nice and light and felt great, or if it was itchy, or if it was kind of rough because you hung it outside and let it air dry. You feel that sensation of the clothes on your skin, but over a couple of minutes, that sensation disappears, even though it's still there. Anybody ever lost their glasses when they're sitting on top of your head? Or been walking around looking for your cell phone and it's in your hand the whole time? This is what's happening. Okay. If a stimulus persists, most sensory neurons do not continue to generate the same frequency of action potentials because their sensory receptors adapt. Adaptation is a process in which the generator potential or receptor potential decreases in amplitude during a maintained constant stimulus. Because of adaptation, the perception of a sensation may fade or disappear even though the stimulus persists. Two really easy real-world experiences that a lot of people go through in their life. Okay, jumping in a cold pool. Right? Or taking a cold shower, hot shower. Cold pool works really well because as kids would always say, get in, you'll get used to it. Did the pool get warmer 5 minutes after you were in there? What's happened? You adjusted, you adapted. Here in Atchison, Kansas, very prominent odor when you step outside sometimes, right? Okay, from the plant downtown, you walk outside this classroom, you'll smell it immediately. 5, 6, 7 minutes go by. Is that odor gone? No, but it is not something you're smelling anymore, is it? Okay. So because of adaptation, the perception of a sensation may fade or even disappear even though the stimulus persists. That's why I had you hold the pen in your hand. You might have no longer felt it. And you could also just do this, like put something like a penny on your arm. You'll feel that penny and then eventually you won't feel it anymore even though it's still on your skin. Now, do all sensations and do all receptors undergo adaptation the same way? No. Okay. We can classify them as rapidly adapting or phasic, slowly adapting or tonic. Rapidly adapting, what we show over here on the right, do exactly what it sounds like. They adapt really rapidly. They generate a burst of action potentials when first stimulated, but even though that the stimulus is still persistent, we see no action potentials till the very end when we have the off response. Examples would be pressure, touch, and smell. We smell it, we touch it, we feel it, but eventually it disappears and it does so pretty quickly. Slowly adapting, what we see is the stimulus is persisting. Over time, our generator receptor potential is decreasing. We're still creating extra potentials, but over time they start to separate out, which means our frequency is going down. But do they disappear? No. Okay. We generate nerve impulses as long as the stimulus persists. Examples would be pain, body position, chemical composition of the blood. Think about those sensations compared to these. Do we want sensations of pain to go away? Absolutely, but only if our body is not experiencing pain anymore. Pain happens when tissues are being damaged. How terrible would it be if you put your hand on that hot stove, you were able to keep your hand on that hot stove so you could, overcome that flexion withdrawal reflex. And then a minute goes by, and then, okay, our brain decides, you know what, that information is not important anymore. And you kept your hand on that hot stove. Exactly. It's not a good thing. Okay? What if we didn't continue to send information to our brain for body position? We wouldn't be able to stay upright. Same thing for chemical composition of the blood. If our pH isn't all right, we got to fix that or we're going to die. So these are like things that we can't just ignore. Make sense? Okay, switching gears now. We've actually kind of talked about this already, but a sensory pathway is a group of parallel chains of neurons convey sensory information from sensory receptors in our periphery to the cerebral cortex. We called these labeled lines earlier. Each of these chains is a labeled line conveying information, sensory information about only one particular modality. The neurons that are part of a sensory pathway are referred to as first, second, third, fourth, or higher order neurons based on which number they are in the sequence or in the chain. First order neurons are the sensory neurons which convey information into the CNS from the receptors. So they're the ones that typically are going to have the dendrites uncovered or covered, encapsulated, carry the signal into the central nervous system, where they then synapse with second order neurons, usually located in either the brainstem or spinal cord. And these ascend to the thalamus, where they synapse with third order neurons. Remember the nickname of the thalamus? You better know it come Tuesday. This is your gateway to the cerebral cortex. They travel here, synapse with the third order neurons, which project their axons to the fourth order neurons in a primary sensory area of our cerebral cortex, where perception of the sensation occurs. Now going back to right here, remember the term decussation? You better remember that for Tuesday. Decussation is called crossing over, which takes place in our medulla oblongata. We're showing that right here. Medulla oblongata is in our brainstem. Now back out here to our cerebral cortex. From the primary sensory area, axons of fourth order neurons extend to higher order neurons and association areas of the cerebral cortex, where complex integration of sensory information takes place. Different sensory pathways extend to and activate different areas of our cerebral cortex. Although most sensory pathways are organized in the manner just described, sensory pathways can differ with respect to the number of neurons in the chains, parts of the brain or spinal cord where they form synapses. So they don't all look exactly like this. The synapsing isn't always right here in the brainstem. Sometimes it's down here in the spinal cord. Most of the time we do travel through the thalamus. Once again, that's our gateway. Most of the time we do decussate in the medulla, but we don't always go to this specific area of the cortex. Sometimes it's different areas, which we already know because we talked about primary visual areas and primary somatosensory areas and that kind of stuff. Okay, so now that we've got really the introductory stuff and understanding all the components that go into sensation, we can actually go through and talk about the different sensations of the body. Not of the body, but our body experiences would be a better way to say it. Okay, so thinking back to our first slide of information, not the title slide, we said that there were two main categories of sensations: special senses, general senses. We said there were two types of generals: somatics and viscerals. So we're talking here on this, obviously, based on the title slide or title of the slide, about somatic senses. Okay, these arise from stimulating sensor receptors in the skin, the subcutaneous layer, which is just deep to the skin, our skeletal muscles, our tendons, and our joints. We've already talked about this, but sensor receptors for somatic sensations are distributed unevenly. In other words, some parts of the body are densely populated with receptors. Others contain only a few. You know, going back to like the tip of our finger versus our upper back. Areas with highest density of somatic sensor receptors are the tip of our tongue, the lips, the fingertips. 4 modalities or 4 types of somatic sensations exist. Tactile include touch, pressure, vibration, itch, and tickle. Receptors involved with tactile sensations include myoclonic corpuscles, which are found in our hairless skin. I thought I had a picture of them, I guess I don't. Within what we call dural papillae or papillae, however you pronounce that. Little ruffle looking like jiggity jaggities in our dermis. Hair plexuses that wrap around our hair follicles. Merkel discs, which are down in the deepest layers of our epidermis. Ruffini corpuscles in the deep dermis. Pacinian or lamellated corpuscles in many places of the body. Thermal sensations involve thermoreceptors, free nerve endings with receptive fields about 1 millimeter in diameter on the skin surface. I don't know why I even really listed that part of it. But there are 2 distinct thermal sensations, coldness and warmth, detected by 2 different receptors. Cold, which are, for whatever reason, located in the epidermis, whereas warm, which are located in the dermis. Cold respond to coldness, warm respond to warmth. Now, the amount of heat and amount of cold they respond to is important because they've got a kind of breaking point where like extreme hot or extreme cold doesn't stimulate them. Extreme hot and extreme cold stimulate what? Pain receptors. Okay. Which, as you can see, are listed down there at the bottom, and as it states, we'll be discussing in the next few slides. Proprioceptive sensations are sensations that allow us to know where our head and limbs are located, how they're moving, even if we're not looking at them, so we can walk, type, or dress without using our eyes. Although using our eyes is very helpful. Okay. Kinesthesia is a fancy word to describe perception of our body movements. And proprioceptive sensations involve proprioceptors. They include the muscle spindles that we talked about last time, tendon organs, which we also talked about last time, and joint kinesthetic receptors, which we have not talked about. We'll actually talk a little more about proprioception after we talk about pain. Okay, pain is a discomfort caused by tissue injury or noxious stimulation mediated by specialized nerve receptors. Once again, we've seen them several times now. Nociceptors. Find them in all tissues of the body except our brain. And they are triggered by chemicals that are released when tissues are injured. Okay. The most prominent and common pain stimulus is a chemical called bradykinin. So when tissues are broken or damaged, chemicals are released, and that's what stimulates the receptors. So these are kind of like chemoreceptors in that they respond to chemicals, but specifically related to pain. Now, when we experience pain, we carry that information to our brain via specific ascending spinal tracts. 2 major ones I want to talk about are the spinothalamic and spinal reticular tract. Spinothalamic tract, as you can see, and I would highlight this, is the most significant pain pathway because it carries most of the somatic pain signals that ultimately reach our cerebral cortex. And it's called the spinothalamic tract because guess where it goes on its way to the cerebral cortex? Using the spinal cord, it travels eventually to the thalamus, spinothalamic tract. We also have a spinal reticular tract, which travels to, guess what? The reticular formation, which is a general area in our brain stem. Talked about that last week, before eventually relaying the information to our hypothalamus and the limbic system, triggering visceral, emotional, and behavioral reactions, like crying, for example, and grunting, and sweating, and blood pressure changes. Remember what our limbic system's known as? It had a nickname. It's not our gateway to the cerebral cortex. That's the thalamus. The limbic system is your emotional brain, because pain triggers emotions as well. Each of us are different in that. Some people don't cry when they get hurt. Some people cry when they get a paper cut. Some people scream. We all experience some visceral changes, though, and behavioral changes as well. All right, there are several ways to classify the type of pain that we experience. Fast pain is a feeling of sharp, localized stabbing pain perceived at the time of injury. We can describe it as acute, sharp, or pricking. I always use an example of like stubbing your toe. Somebody actually, I think, said that already today. Stubbing your toe hurts. We have all experienced it. We've all said the cuss words that go along with it. You know exactly where you feel that pain. It's in your toe, right at the tip of your toe. It hurts, and it's immediate. That signal can travel via myelinated pain fibers at speeds of 12 to 30 meters per second. That's an incredible amount of speed. After a minute or two goes by, that pain starts to kind of disappear, but it might feel now like your whole foot hurts, and it might be kind of pulsating. You guys know what I'm talking about? That's what slow pain is. It's longer lasting, it's dull, it's diffused, still travels fairly quick. Unmyelinated pain fibers, 1/2 to 2 meters per second. It's what we feel like after a long day of standing up, working in the yard, whatever. You know, it's a chronic, dull, achy pain. You know, I always say my dogs are barking after a long day. I love that phrase. Okay. It's a throbbing type pain as well, we can describe it as. We can also describe pain based on where it's coming from. Superficial somatic pain is pain originating from our skin, whereas deep somatic pain is deeper. It's in our skeletal muscles. It's in our joints. It's tendons or fascia. There's also visceral pain, pain from our viscera. Once again, viscera refers to your internal organs. So like, you know, gallbladder pain when you have gallstones that Jennifer was talking about. All right, I talked about the kind of first bullet there. Fast pain is very precisely localized to the stimulated area. For example, if someone pricks you with a pin, you know exactly which part of your body was stimulated. Somatic slow pain also is well localized, but more diffuse, meaning it involves larger areas. You know, you might feel like the whole muscle hurts instead of just one specific part. In many instances of visceral pain, pain is felt actually not within the organ itself, but within the skin that overlies that stimulated organ. We call this referred pain. And it occurs because the brain assumes visceral pain is pain coming from the skin, when all actuality it's coming from something like your heart. And it's not really a complicated reason as to why. It's pretty simple, and it's shown here in our figure on the right, where we have two first order neurons, one coming from the skin, one coming from the heart, traveling into the spinal cord, and synapsing with the same second order neuron. And that second order neuron then carries a signal up the same way it would if it was coming from the skin as it would if it was coming from the heart. So our brain doesn't really know if it's coming from the heart or from the skin, and it's just going to assume it's coming from the skin. This is what happens when somebody has a myocardial infarction, which you may know best as what? Heart attack. Anybody ever heard that if a person's having a heart attack, they are going to feel not only chest pain, but pain down their left arm. The figure on the left side of the page actually shows you a mapping of various referred pain sites. Some of them are, fairly straightforward. Your kidneys aren't too far away from where they're mapped out. What's crazy is some of them, like you can actually feel jaw pain during a heart attack. Gallbladder pain up in your shoulder. Weird. It's kind of super weird. Okay, proprioception. Once again, this is what allows us to know where our head and limbs are located, how they're moving, and if we're not looking at them. Allowing us to walk, type, or dress without using our eyes. We already had all that information a couple slides ago. It's just an exact thing. Kinesthesia, we already said that's the perception of body movements. And receptors involved are called proprioceptors. Proprioceptors are found in our muscles, especially our postural ones and tendons, where they provide information on the degree to which muscles are contracted, amount of tension on tendons, and the position of joints. These were the ones that we talked about the other day with the stretch reflex, with the tendon reflex. When you tap on the patellar tendon, that's the stretch reflex, so on and so forth. One cool thing they do is allow us to perform weight discrimination. Ability to assess the weight of an object is especially helpful with determining the muscular effort necessary to perform a task. When you lift a box, when you go to pick it up, it looks super heavy. When you go to pick it up, anyway, ever experienced this, and it's super light. Okay, it's almost like your whole balance gets thrown off because you put in a lot of force when you didn't really need to. We're never going to lift, at least we don't have to, something light with maximal effort like this, reminder. I'm not putting much effort into lifting that up. Okay, I don't require a lot of effort to lift a light box up. Okay, that's what weight discrimination refers to. It's really, really helpful because it keeps us from getting tired really easily. If you're putting maximal effort to lift everything up and do any type of activity, you're going to experience fatigue pretty quick, right? So it's preventative against that.
Is that kinesia?
Kinesthesia?
Is that that's what they would study like a physical therapy school, right? They have like kinesiology or something.
Yeah, kinesiology.
That's what it's talking about, right?
Kinesthetics. Kines refers to movement. Like my degree was in kinesiology, or both of them are in kinesiology. It's just the science of body movement.
I thought that might be what that was, though, when it.
Kinesthesia refers to perception of body movements.
Akathisia?
Like the esthesia, esthesia, like that part of it. Yeah, probably it just refers to sensation of some sort. You could probably Google it and find out. I was talking earlier about that synesthesia that some people experience, where they can like taste colors and that kind of stuff. I'm assuming aesthesia refers to sensation. Following our examples of the slowly adapting proprioceptors found in our joints. Really, joints is kind of a bad way to say it because muscles aren't joints, but they're associated with joints, where you find your muscle spindles. Muscle spindles are proprioceptors in our skeletal muscles, monitoring changes in our length of skeletal muscles, where they participate in stretch reflexes. By adjusting how vigorously a muscle spindle responds to stretching of the skeletal muscle, our brain sets an overall level of muscle tone. Once again, we said it is a small degree of contraction present even when a muscle is at rest. Tendon organs, as are listed here, we also talked about them the other day. Golgi tendon organs, located at the junction of a tendon and a muscle, are involved in tendon reflexes and act to protect tendons and their associated muscles from damage due to excessive tension. Joint kinesthetic receptors, which are really brand new to us, found around the articular capsule of our synovial or most major joints, responding to pressure, acceleration, and deceleration of joints during movements, and adjust muscles when excessive strain is placed on the joint. I only included the illustration here because it shows, here's a set of tendon organs at the tendon of this muscle. Surrounding our muscle fibers, you can see some sensory muscle spindle fibers. So, all right, let's talk somatic sensory pathways, which relay the information from those various somatic sensory receptors just described to a specific region in our cerebral cortex called the primary somatosensory area. We also send some signals to our cerebellum. Okay. The pathways to the cerebral cortex consist of thousands of sets of 3 specific neurons. The first order neurons are the ones that conduct the information from the somatic receptors in our brainstem, into the brainstem or spinal cord, either traveling via cranial nerves into the brainstem or spinal nerves into the spinal cord. From there, second order neurons carry the signal up to the thalamus. They are the ones that decussate or cross over in our brainstem or spinal cord before ascending to the thalamus. And then from there, the third order neurons are the ones that go to the somatosensory area of the cortex on that same side. Somatic sensory impulses ascend to the cerebral cortex via 3 general pathways: a posterior medial lemniscus pathway, anterolateral pathways, and the trigeminal thalamic pathway. Not that that's super important. Last thing on, I believe it's last thing anyway, on somatic sensations is that primary somatosensory area. And what we have here shown is a funky figure. What this illustration is showing you is a mapping of that specific area of our cerebral cortex, what we call the primary somatosensory area or primary somatosensory cortex. To demonstrate that, for example, our face and our hands dominate a very big portion of that portion of our cerebral cortex. The bigger the area, really the more sensitive it is. Primary somatosensory area occupies the postcentral gyri of our parietal lobes of our cortex. And, for example, our genitalia, when we stimulate those, that's where that goes in our primary somatosensory area. The tongue's down here, the thumb's up here. What's shown here is actually called a homunculus, specifically a sensory homunculus, which, as you can see here, but oops, I did not mean to click on that. You can actually buy models to mimic the homunculus. I always show this. It's kind of a weird, creepy thing.
Yeah.
People buy it. And actually, last semester I was showing this, and a person thought it was so cool that they went out and looked for one themselves to buy. But
It's interesting.
very interesting. It's a decent way to explain it, I guess. Okay, this seems like a very great spot to actually stop I know we're not quite at 1:30, but that's okay because we got a lot accomplished and we're ready for about the last half of the lecture. Really not quite that much, but we'll go through the 5 special sensations next week.